callosum, the hippocampal commissure, and the anterior commissure, right? And you can roughly get an idea of the proportion and in size of these commissures in this tenrec. And one way to understand why this is probably because the tenrec brain is mostly neocortex. Sorry, it's mostly piriform cortex or olfactory cortices. So in a number of basal placental mammals, most of the cortical areas are not the neocortex or isocortex, but roughly piriform or olfactory. So here you can see on the left at the bottom an armadillo, and you can see a huge piriform cortex that's also gyrencephalic, as well as olfactory tubercle and other areas. And the rhinal fissure that is shown here with letters RF is actually indicating the limit between the paleocortex or piriform cortex and the neocortex or isocortex above. So whereas most of the corpus callosum would carry isocortical fibers, the anterior commissure would carry fibers mostly from the piriform cortex in this case. So this is an example of a huge diversity in the proportion of sizes roughly between the corpus callosum and the anterior commissure. Other animals, such as the elephant shrew, shown here on the right, they also have a tiny corpus callosum that you can see there as a very thin band crossing above the hippocampus. But what they do have is a big piriform cortex as well, but also a massive hippocampus, right? So this animal relies a lot on spatial orientation and finding their burrow system in a very complex underground system of tunnels. So the hippocampus is massive, and so is the hippocampal commissure of this animal. So the hippocampal commissure is even larger than the corpus callosum. So basically, it's really important to also consider the ecology of animals and their lifestyle to get a better idea on the anatomy and what's the functional relevance of comparisons between different brain areas. Right, so the first person who discovered that not all mammals have a corpus callosum was Richard Owen. Okay, during the times of Darwin, he's pre-Darwinian. He was anti-evolutionist in a way, but at the same time a comparative anatomist. He's the one who coined the word dinosaur, and he discovered that the brain of wallabies, shown here on the right, they don't have a corpus callosum, and instead they had this extended anterior commissure, or expanded, a larger anterior commissure. And he also described this hippocampal commissure as probably serving the functions of the corpus callosum. So it wasn't at that time very clear what it all meant. The techniques at the time weren't appropriate to elucidate the circuitry of connections. So this was mostly macro anatomy. And then a few years later, William Flower discovered that this situation also extended down to platypuses and monotremes. So if you look at the echidna brain as well, they also lack a corpus callosum, and instead they have a huge anterior commissure, okay? But it wasn't until Grafton Elliott, an Australian-born neuroanatomist, who discovered, and using more detailed anatomical techniques, discovered that actually only the placental mammals have the corpus callosum, and non-placental, such as marsupials or monotremes, they have an expanded anterior commissure, which carries most of the connections between hemispheres. He also made a very interesting finding that is really interesting to me and many people in the field as well, that there is an extra tract shown here on the right with the letters FA that is called fasciculus aberrans, that only exist in a lineage of marsupials that belong to the wallabies and the koalas, known as the diprotodon marsupials. And they have this bundle of fibers that connect the dorsal part of the neocortex through the anterior commissure, but instead of going all the way down the external capsule, these fibers would follow the internal capsule to join the anterior commissure on its more dorsal domain. So this is a very interesting case. There's no other mammals that had shown this interhemispheric connection other than the diprotodon marsupials. So it's a really interesting system of study to understand what goes on during axon guidance and development of this circuitry, because pretty much these commissural axons would be taking the subcerebral route, right, the internal capsule, but then exiting that route to join the anterior commissure and then project to the contralateral hemisphere. So you can see this was towards the end of the 19th century, and roughly at the same time, Cajal started doing very detailed Golgi stains and neuroanatomy reconstruction of all these systems. And he was already recognizing the different components of the corpus callosum and the anterior commissure, which neurons project through those in different planes of sections and in many different animals. So we can truly consider him as the father of systems neuroscience and circuitry. So one of the very early observations that Cajal did was that the anterior commissure of rodents, as we know, would carry axons from piriform cortices. There's also axons that come from more neocortical components, or at least regions that lie above the rhinal fissure that would correspond to proper isocortex. So at the time, it wasn't really well known what that all meant. And even today, we don't have a lot of studies with enough anatomical resolution that would distinguish whether there are neocortical axons that would project down the anterior commissure, and let alone whether that would be a different subpopulation of neurons that would project through the corpus callosum, or whether there could be some neurons that would extend axons that could branch to both the corpus callosum and the anterior commissure, which would also be a very interesting scenario that hasn't been fully elucidated at the time. Okay, so this Grafton Smith neuroanatomist guy had a lot of insight and a lot of interest in development. So he compared many different species, and you can see here the date 1897. And by that time, he was already proposing that if we look at the midline in the sagittal view, so here we can see the reptiles having the anterior commissure more ventral, so this would be a sagittal representation of the midline, and the hippocampal commissure immediately below, above that, sorry, dorsally. He realized that in mammals, there was this massive expansion of the anterior commissure as well as the hippocampal commissure. And he kind of proposed a progression or a developmental series that could explain the evolution of the corpus callosum, basically by extension of this midline component more dorsally, roughly near the area of where the hippocampal commissure actually forms. So he also looked at what he called lower placentals, such as the hedgehogs or armadillos, species that I was telling you before that have a tiny corpus callosum. And he was basically proposing that there could have been some sort of an extension of this territory, and that fibers that would otherwise cross through either the anterior commissure or the hippocampal commissure would now start crossing the midline into more dorsal components to actually form the corpus callosum proper, as we know, in higher placental animals such as primates or carnivores. So this was a really early insight, and it paved the way for many subsequent developmental studies on the evolution of the corpus callosum. So now we can see that, for example, if we look at the mouse brain stained here with Niesel, and we take three horizontal sections across different levels. So the most ventral level would be across the anterior commissure, shown here on the top panels. So you can see the anterior commissure, the rostral branches joining to the posterior branches here on at the bottom. Then we have a middle section, roughly at the level between the commissures, and a more dorsal section shown here at the bottom, where we have the corpus callosum at the front and the hippocampal commissure at the back. One thing that's been known in the comparative anatomy literature is that the septum, particularly the midline of the septum, is fused. There's no space between the both halves of the septum across the midline in all of the eutherian or placental mammals that have been examined, but that may not so much be the case in non-placental mammals. So therefore, there was this idea that the fusion of the septum could have something to do with the evolution of the corpus callosum. So later, people started discovering glial populations such as the midline superglia, work pioneered by Jerry Silver, that would do experiments in different animals to actually show that that would be the process. And this is part of the work that was pioneered by Ilan Gobius in our lab, that was actually trying to elucidate what was the molecular pathway that is involved in this process of remodeling of this midline territory. So here in the left, we can see horizontal sections of the embryonic mouse brain at embryonic days 15, 16, and 17. And you can see that in this horizontal plane of section, there is the interhemispheric fissure, which is actually the space between hemispheres. Then there's the third ventricle at the bottom here that is squished. And this space in between there is glial cells that actually remodels and fuses this whole territory to actually closing the space at the level of the septum. And this glia will be then intercalating at the midline, acting like a zipper, and at the same time producing guidance cues and also a substrate that would help callosal axons to cross the midline. So this has been also associated with callosal agenesis in humans. So in this study in 2016, there was a lot of human patients that failed to form the corpus callosum. They also show like a separation in these septal halves. So in the schematic on the right here, we can see the scenario that during normal human development, callosal axons are about to reach the midline, and the radial glia at the midline has still not fully intercalated to fuse this territory. And there is a molecular pathway that is triggered by FGF8, following the MAP kinase signaling cascade to activate the nuclear factor 1 family of transcription factors, A and B, that would then promote an astrogliogenic program, shown here with GFAP, that would signal a transition of these radial glia into multipolar astrocytes that would then migrate towards the midline and fuse this territory, thereby producing a substrate for callosal axons to form. And if you interrupt this pathway at any of these stages, then the multipolar astrocytes fail to actually fuse this midline, and the callosal axons cannot cross the midline, and then we have these props bundles. So this would be a model of one of the perhaps most relevant events that are required for callosal development. So then the question was, well, what if we look now at non-placental mammals, such as the platypus or marsupials? So we took brains from the platypus and put them in the scanner. And if we look at the horizontal plane as well, we can see that the interhemispheric feature remains separated throughout ontogeny. So if we look in an adult, there's no fusion of this septum territory. And similarly, in a marsupial, our Danards, we also find the same, that the septum remains separated and there's no actual fusion of this territory into adulthood. So a question that comes is whether and how much this is actually linked to the evolution of the corpus callosum. So if we know that this midline tissue remodeling only happens in eutherians, how did that evolve? What actually happened in the first time it did happen, and how that was conserved during evolution is an interesting question that may take years to actually understand what are the mechanisms related to the onset of this fusion mechanism. Okay, so now if we go a little bit back into vertebrates, including all of the tetrapods, we can see that the telencephalon is connected basically by two main commissures. One being the anterior commissure, as I was telling you, is present in all vertebrates, connecting mostly olfactory recipient structures. And right above that, there's the hippocampal commissure, which is actually called hippocampal commissure only in mammals, because in mammals we have a proper hippocampus, but in non-mammals it's also called the pallial commissure because it's connecting the medial pallium, which would be the homologous area to the hippocampus. So this pallial or hippocampal commissure is also present in frogs and then in amniotes such as lizards and pigeons. And then in mammals is where we have this huge expansion. First, with the origin of the neocortex or isocortex, that would also have entail an increase in commissural pathways between the cortical hemispheres, and then subsequently later by the evolution of the corpus callosum exclusively in placental mammals, where we can see here an example of the tenrec, that they have a huge piriform cortex and a very tiny neocortex, therefore their corpus callosum is tiny, and another species such as mouse or humans as well, where the neocortex is larger than the olfactory cortex, and therefore the corpus callosum is also much larger. So these are myelin-stained sections through the sagittal plane of the brains of mammals and a bird as an outgroup here, the zebra finch, okay? So basically I wanted you to have a look and compare the sizes of the commissures. So in the zebra finch, you can see the anterior commissure, very tiny there, that spot, and the hippocampal commissure immediately above as very tiny. But if we already look at a monotremes such as the echidna, we have a huge expansion of the anterior commissure. The hippocampal commissure as well is large, located right above. Here's another marsupial, the Tasmanian devil with the big anterior commissure and the hippocampal commissure, and then the human for context, where you can see the huge corpus callosum and a very tiny anterior commissure, which is tiny, and we know that humans and all primates have very small olfactory areas of the brain as well. So if we now have a closer look to the connections in the birds to get an idea of how things might have been before the evolution of mammals, we can see the panels on top here. This is the coronal view across these commissures. So we have the anterior commissure there and the hippocampal commissure connecting the areas that are homologous to the hippocampus, that are in the medial pallium, shown here with the HP for hippocampus and the area parahippocampalis or parahippocampal area here. You should also know that the hippocampus in non-mammals is not layered in a way as we know the mammalian one to be. So instead of having this shape, it's just like nuclei. But the circuitry is fairly homologous, and the functional roles is also very similar concerning memory, episodic, as well as place, spatial memory and location. Okay, so at the bottom here, we can see horizontal panels cut at two different levels, one being more ventral at the level of the anterior commissure, another one a little bit above to show the hippocampal commissure. And although we can't really get a full idea of the connections here, things that we can already realize is that the homologous areas of the neocortex in birds are not layered, okay? So we understand the neocortex as the 6-layer organization that's only present in mammals, but birds and reptiles instead are organized in nuclei or clusters of neurons. Shown here, you can see very clearly in the horizontal plane with these letters M for mesopallium, the nidopallium is N, E is entopallium, and the arcopallium at the more caudal portions here in A. So all of these areas are somewhat equivalent to different layers. There's still a huge debate in the field as to which areas of the non-mammalian brain correspond to the neocortex, whether there is a field homology to different areas or whether different layers of the cortex would be homologous to some of these nuclei. But one thing that was recently studied and published in 2016 by Letzner in the Honor Gorkuntum group from Germany was that the connections in the pigeon between hemispheres, while we know the hippocampal connection is very simple, so in brown here, we can see the hippocampal area connecting through the dorsal portion of the commissural pathways. The anterior commissure, on the other hand, is mostly arising from the one area called the arcopallium at the back of the brain, as well as the amygdala area or amygdaloid homologues in the pigeon. So those will be the main areas where commissural neurons will arise from, and then the axons cross the midline to terminate in several different areas. So instead of being a homotopic connection from many areas to many targets, as we know in humans, or in mammals to be the interhemispheric cortical connections, in birds, most of these interhemispheric cortical-like connections arise from one or two few areas, and then they would terminate in several different targets in the contralateral hemisphere. So that's already a huge difference in the logic of the circuitry between hemispheres. Okay, so we have this situation here on the left at the bottom of the pigeon where connections from the arcopallium in one hemisphere would cross the midline and project and terminate into many different areas of the contralateral hemisphere. Whereas in placental mammals such as humans and mice, we know that most areas of the neocortex as well as the piriform, they would cross the midline in a segregated manner where axons would be arranged along the tract in a position that would actually recapitulate both the position of the cell bodies as well as the position of the terminals. So in a way, you can predict where these connections are connecting based on the position of the axons within the tract, right? So we have that segregation of fibers along the tract. So a question was, how would this situation be in a non-placental mammal such as the platypus or the marsupials? So they do have a neocortex, they do connect through the anterior commissure, but are these axons also segregated by the position of the cell bodies? Are they also connections that would go from one area to homotopic areas in the control hemisphere, or would instead be more similar to a non-mammalian connection that would go from one area to several other areas? So in order to investigate that, we have a look at the brain of the platypus, and we put a small ROI in a DTI reconstruction inside the anterior commissure in 3 planes. So first we will move this ROI in dorsal to ventral, and then anterior to posterior, and then again in a diagonal fashion. And what we can see is that the reconstruction of these tracts very beautifully follow the position of the ROI. So we can basically see the segregation of fibers in this Cartesian plane of going dorsal ventral, as well as anterior posterior, as well as diagonal. We can actually see the reconstruction that would be following this pattern. So when we put all of these tracts together, we can see that in the platypus brain, fibers coming from the piriform cortex, they will cross the midline more rostrally. And then if we have a look at neocortical components, here shown in orange, yellow, and green, they also would cross the midline in very segregated ways. Here we can see the entorhinal cortex from the back of the brain. And on the panel B here, we can see a zoom-in version of this is a reconstruction of tracts using DTI tractography and hardy statistical reconstruction of these fibers. So this must always be taken with a grain of salt, but this is still 16.4 Tesla, so it's high resolution. We can be fairly confident that there is some, this is an actual representation of what's going on in the animal. So then as a proof of concept of this, we can now have a look at the midline plane of the platypus brain. So that's the anterior commissure there. And we can ask the software now to show us all the tracks that would cross the midline through these different domains that we would now call piriform domain, the rostral part of the neocortex, somatosensory primary areas, and entorhinal cortices. And just by doing this parcellation at the midline, we can reconstruct most of these tracts in a segregated fashion as well as homotopic fashion. So it's actually indicating that the main features of callosal connectivity that we know of today were already present in a very ancient mammal such as the platypus. So here's again a 3D reconstruction of all these fibers, showing this concept of axonal segregation as well as homotopy between similar areas in both hemispheres. So we did the same with Danner brain. So this is our marsupial. And we actually here came and drew regions of interest manually inside the anterior commissure that would actually recapitulate different components of interhemispheric connections. So we can see here the olfactory component in yellow on the right. Then we have frontal areas, that's the forceps minor equivalent that goes to the prefrontal areas, cingulate cortices here shown in orange, neocortical and entorhinal. So it's basically the same concept. And as another proof of principle, if we draw our eyes within S1 at different parts of the primary somatosensory area, such that would correspond to the hind limb, the mandible, and the mestacial whiskers, we can then see axons that would go down both the external capsule to cross the midline at the anterior commissure, as well as the internal capsule. So those are the descending or ascending axons, because we can't tell direction. But we can already see that this segregation of fibers is conserved in both the internal capsule as well as towards the anterior commissure. As they cross the midline, shown here in panel D, we can see an orthogonal plane showing the hippocampal commissure above. And as these fibers cross the midline into the opposite hemisphere, we still can see this arrangement, right, from a lateral to medial proportion, suggesting that there is this topographic segregation of fibers that is conserved. So this is a 3D reconstruction again of the Danner brain, showing very beautiful arrangement of how these fibers segregate along the midline and how they terminate into homotopic areas in the contralateral hemisphere. So to check that this was actually a real thing, we also did injections of dye in the fixed tissue and then keep the brain in the oven for a few months to give time for these dyes to diffuse. So when we put different dyes in a more medial or lateral components, we can see that these axons are actually segregated along the midline at the level of the anterior commissure. And we do the exact same experiment, but now injecting more rostrally in the anterior olfactory nucleus, as well as in the S1 area. We can again reconstruct the rostral part of the anterior commissure, shown here in green, as well as the isocortical component, suggesting that this is actually a real segregation of axons. So then we did injections in the stereotaxic frame to actually map the circuitry of connections in the donor. And one of the main things that we found is that there is homotopy. So say if we inject, for example, in this area in red, we can see the cell bodies in the contralateral hemisphere in a roughly similar areas. So that you can see, for example, here in yellow, you have cell bodies in yellow in a similar area there. Same if you go very caudal towards the retrosplenial cortex, you can see homotopic cell bodies in corresponding areas. But something that caught our attention dramatically is this region here known as the claustrum, where you can see lots of dots in blue and light blue and red, right? So the claustrum is also is non-layered cortical structure that it lies immediately under the cortex at the level of the spinal fissure, and is known to be a hub of cortical connections both within hemisphere as well as with the contralateral hemisphere. So here's an actual experiment with the injection site shown there at the back of the brain, where we can see homotopic cell bodies in the contralateral hemisphere, but also a very dense collection of neurons inside the claustrum that will be projecting to that area, okay? And not only that, but also the claustrum has a topographic arrangement inside this nucleus of neurons that will be projecting to medial lateral parts of the contralateral cingulate cortex. So this is a very interesting hub of connections that will go from this tiny little structure that's hidden between cortices, and then project to the same as well as the opposite hemisphere in a topographic manner. So with donors, we have this advantage that they complete their development inside of the pouch. That's my hand there holding a donor. So they're tiny, they're smaller than a mouse. And we can just open the pouch and see this animal developing. So at postnatal day 10, we can electroporate their brains with TD tomato. And this would be the stage equivalent to deep layer neurogenesis, embryonic day 12, roughly. And then we can come back and in the same animals now electroporate a green protein 10 days later, equivalent to embryonic day 15 when the upper layer neurons are being born. And actually what we can do with this is very simple protocol to label the different layers that would be projecting, in this case, down the internal capsule for the deeper layers, as well as contralaterally for both deeper and upper layer neurons. So this is very hard to do in mice because you have to open the mother up to do in utero electroporation. So it's very invasive. So you want to come back 3 days later, there is a lot of mortality and it's a mess. And also the anatomical resolution is limited because you have the uterus as well as the amniotic sac as barriers between your electrodes and the actual developing embryo. So it's very hard to get anatomical accuracy. Whereas with donors, we can use these tiny electrodes and you're directly contacting the skin and the jaw. There's no uterus, no amniotic fluid to be messing in with the accuracy. So this actually gives unprecedented accuracy to transfect many different parts of the brain. So in this case, we're just transfecting pretty much anywhere that you want in the brain. You can, in this case, overexpress fluorescent proteins. So by doing this technique, we then did anterograde mapping. So we would be co-expressing a fluorescent protein in one hemisphere and then have a look at the targets in the contralateral hemisphere. So one of the main things that we found is that there is homotopic connections. So cell bodies in here, for example, in the middle palm, they terminate in homotopic similar areas, but there is also heterotopic terminations that are mostly confined to the medial and the lateral portions of the neocortex, okay? So basically the concept here is that pretty much everywhere in the neocortex that you would electroporate, you would see terminals in a homotopic area, but also in heterotopic regions of the medial and the lateral parts of the brain. So this is the situation in the donor. We don't know if these connections would be because of these neurons are branching or they're intermingled. So to elucidate this, so I put there a question mark. we use two different dyes, retrogradely labeled. So we would inject in green in one hemisphere, I mean in the medial part, and then lateral with a red one, and then have a look at these medial and lateral parts of the neocortex to get an idea of how many of these neurons would be projecting exclusively to one or to both of these areas, and whether these are heterotopic, meaning a different connection, or homotopic, meaning the same. So basically what we find is that in the medial part of the cortex, we see both cells in red and in green, whereas in the lateral portion, we have mostly connections that will go to the homotopic target. So in this case, it's red. So the homotopic connections are mostly represented in the lateral parts than in the medial parts, whereas the heterotopic connections are significantly more abundant in the medial portions of the neocortex than the lateral ones, suggesting that it's the cingulate or medial dorsal parts of the cortex are highly hyperconnecting heterotopic regions. So these projections also mirror the ipsilateral projections. So we often focus on the contralateral circuit, but these neurons are also projecting to the same hemisphere. So in this example, we electroporate the perirhinal cortex and we can see contralateral axons terminating in the homotopic area, but also here there's axons terminating in the ipsilateral claustrum as well as the contralateral claustrum. So basically pretty much the same patterns that we find in the same hemisphere, ipsilateral, they have a similar representation, albeit with less axons in the contralateral hemisphere. And this is a similar thing whether we electroporate in S1 and then we have a look at the cingulate cortices shown here, or in this case, these axons are avoiding the claustrum. So you can see the claustrum dotted here in the bottom panels in the ipsilateral hemisphere as well as the contralateral one. So basically what we find is that there is a pan-mammalian connectome that likely arose more than 80 million years before the corpus callosum, and that is made of contralateral homotopy, axonal segregation at the level of the tracts, as well as hyperconnected hubs in the medial and in the lateral portions that include the insular as well as the claustrum region. Okay, so the final part's got to do a bit more of the genetic arrangement and what's going on with the axon guidance mechanisms. So basically, we know that different layers of the cortex will have different targets, with the upper layers being mostly intracortical, including the callosal component, deeper layers being mostly corticothalamic, as well as a huge component of layer 5 that would project subcerebrally down the spinal cord, as well as the tectum and thalamus. So these three main populations of cells are also controlled by different transcription factors. So here I'm highlighting SATB2 and CTIP2 because we know that they interact to direct callosal neurons. In the case of mouse, we know a lot about the role of SATB2 in specifying callosal neurons, and CTIP2 that would be specifying subcerebral neurons or corticofugal projections. So from the literature, we know that SATB2 inhibits CTIP2, and that CTIP2 would project laterally to then join the internal capsule, where SATB2 positive neurons project medially in the mouse to form the corpus callosum and connecting to the contralateral hemisphere. So studies in the SATB2 knockout animal have shown that this animal, they don't form a corpus callosum, and axons that would be callosal, they instead go either down the internal capsule or down the external capsule into the anterior commissure to terminate into contralateral targets. So this was very exciting because it resembles the phenotype of marsupials, suggesting that SATB2, which would be important for forming the corpus callosum, if you knock it down, then you have something very similar to the marsupial, where interhemispheric connections are preserved and they will go down the anterior commissure. So the big question is, what would SATB2 expression be like in a donor, right? So the easiest thing would be, oh, maybe donors, marsupials, they don't have SATB2 and that's why they don't have a corpus callosum. But it's not that simple, right? So to get a better idea on that, we did retrograde injections, in this case in the colliculus, to label subcerebral projections in layer 5. And we found that all of these subcerebral projections in the mouse, they all express CTIP2 either without or with SATB2 as well. So these are the different proportions in blue and purple CTIP2 positive neurons that project subcerebrally. In the donor, they also express CTIP2, more than 97% of them are CTIP2 positive, either without SATB2 or with SATB2 as well. And if we inject in the contralateral hemisphere and then we focus at the commissural neurons in layer 2-3 as well as layer 5A, we can see that in the mouse, most of these neurons express SATB2 without CTIP2, and a few percentage don't express SATB2, which probably include interneurons. Whereas in the donor, the vast majority of these neurons that project contralaterally do express SATB2, right? So then SATB2 is not a callosal specifying gene, but it's rather, you can think of it as a commissural gene, right? Because it observes the similar connections in animals that didn't evolve a corpus callosum. So when we look at development, one thing that we found is that CTIP2 starts to be expressed, shown here in green, in similar stages and in similar layers as the mouse, as well as donors. But SATB2 appears relatively later in development in the mouse than they do in the donor. So if we look here at stage 20, which would be postnatal day 8 to 11 in the donor, or embryonic day 12 of a mouse haven't started expressing SATB2 yet, but donors are already expressing high levels of that. So that's something that we kept in mind. And we wanted to know whether if we can ectopically overexpress CTIP2 now in the upper layer neurons, would that be sufficient to promote a subcerebral projection fate? And whether if we take the gene from the mouse and put it in the donor, would it have a similar role? And if we do the same experiment? So this is a control where we electroporate upper layer neurons and we see axons terminating in the contralateral homotopic area. There is no axons going down the internal capsule here. So those are just corticostriatal terminals. But when we put the mouse sequence of CTIP2, we now see a huge projection down the internal capsule that would terminate into thalamus as well as the colliculoid. And interestingly, if we put the Dannard sequence, we see exactly the same, that now these axons show a huge projection that's descending. Now if we do the similar experiment in the Dannard, so we put the control, we see no axons going down the internal capsule. But if we put the mouse CTIP2 sequence, we now can see these axons that we also see terminals in the subcerebral targets. And if we put the Dannard construct, again, we see a huge increase in this subcerebral pathway, suggesting that CTIP2 has this conserved role of specifying a subcerebral projection fate. So then we did a CRISPR approach where we actually designed two different guide RNAs to introduce mutations in the SATB2 gene in conserved regions between CAT1 and CAT2 areas of the SATB2 gene, and electroporated this into the mouse as well as in the donor. So what we found here is that as compared to the control, if we silence SATB2 now in the donor, that's also sufficient to promote new ectopic projections down the internal capsule. And if we do the same procedure in the donor, again, just by silencing SATB2 from the upper layers is sufficient to promote subcerebral projections, suggesting that either overexpression of CTIP2 or silencing of SATB2 can promote these descending cortical projections. So then we wanted to see what happens now if we put SATB2, but at the early stage, right? So in the mouse, if we electroporate early, we see also descending projection, but we can also see contralateral projection from deeper layers, layer 5 neurons mostly. If we now put SATB2 ectopically at this early stage, we see a huge increase in the anterior commissure component, which was a little bit puzzling at first because we knew that if you knock it out from the mouse, you have an increase in the anterior commissure, but now if we overexpress SATB2 as well at the early stages, we also see an increase in commissural fibers going down the anterior commissure. And the same happens if we now put the Dannard sequence of SATB2. We also see this increase in the anterior commissure pathway. So what could that be? And then if we do the same equivalent experiment in Dannard, we see no difference, right? The Dannard, whatever we do, we see similar amounts of axon going the anterior commissure. So one possibility is that it's because of this difference in the timing of SATB2 that could be doing all of this. So basically, if we look at SATB2 expression at stage 19, we can see that neither mouse nor donors have a lot, but by stage 20, the donor already has a lot of SATB2, right? That's stage 21. We start to see a little bit in donor, but in mouse, sorry, but still much higher in donors. And only by stage 22, like 2 stages away, we start to see an increase in SATB2 expression, but it's still significantly less than donors. And only by stage 23, they match up to similar levels. So maybe the experiment that we were doing, rather than increasing SATB2 at the early stage in mouse, we were increasing it at a stage when it was very low. So in this experiment, when we electroporate SATB2 at stage 20, it's probably because there is so little SATB2 being expressed at that time that we're forcing the system to either mature or to trigger an axon guidance program, that this axon would then encounter the environment that's already there, probably telling them to cross the midline through whatever substrate. In this case, the anterior commissure is forming and it's already developed at an earlier stage. So that could be one way where these axons could be doing the targeting. So then the hypothesis was, what if we now electroporate the same SATB2 at a stage when they already have large levels of SATB2 expression, and then we see no effect, suggesting that it's got to do with the timing of this SATB2. And the fact that the donor is expressing SATB2 at such earlier stages compared to mouse is an interesting possibility of things that could be triggering the evolution of these pathways. So in conclusion, we find that CT2 has a conserved role in promoting the subcerebral fate, and SATB2 would be repressing this subcerebral fate. And that is similar throughout therians, regardless of whether they have a corpus callosum or not. And that main question, could it be that differences in the timing of expression of regulatory networks, in this case SATB2, or what we know as heterochrony, or difference in the timing of transcriptional control, could those differences be sufficient to generate evolutionary new pathways, such as the corpus callosum? So this is a very interesting topic in the field of evo-devo, how by just switching subtle differences during development, you can have a huge effect in phenotype, possibly by generating evolutionary innovation in complex traits. So that's all that I wanted to tell you today. I'd like to acknowledge and thank you very much, Professor Linda Riches, who's been a wonderful mentor, as well as all the people that has contributed to this work: Laura Fenlon, Annalise Paulino, Peter Kosulin, Ilan Gobius, Laura Morcon, Tim Ewers, and Newman Kurniawan, as well as all the funding bodies. And here's my contact details if you have further questions, and I'm happy to take questions now. Thank you very much.
Thank you, Rodrigo. It's a tour de force of closal evolution. I'll just remind everybody, in case you missed at the beginning, the protocol for questions. If you'd like to speak, I will need to click a button on my end to allow that. So just either type your question in chat and I can direct Rodrigo to it or read it out for you, or let me know in chat if you would like to speak with your microphone and I can make that happen. Okay, no questions yet. Well, I'll kick off with a question that, oh no, Benaz has a question. Okay, so Benaz should be able to talk.
Yes.
Yeah, go ahead, Benaz.
Yes, hi. Thank you very much, Rodrigo, for this very, very interesting talk. So I have a question about the elephant shrew. You show coronal section in one of your slides, and I was impressed by the size of the hippocampus. Right, they had very large hippocampal regions. So they have corpus callosum, or they have very thin corpus callosum, the elephant shrews. And what about the hippocampal commissure? Is it then thicker because the hippocampus is very large?
Yes.
And how is hippocampal commissure then connected to the anterior commissure? Are they developmentally related? Can one develop without the other one? You know, what is the
Right. So
relationship between
It's very hard to know because it's hard to study this species. It's very, very elusive and it's in danger. So there's not many mechanistic studies on its development. But yes, they do have a corpus callosum because they're placentals, they're eutherians, but it's very thin. So you can see in the coronal section a very thin corpus callosum. The hippocampal commissure is massive. So you can see sitting right under the corpus callosum, there is this massive hippocampal commissure, whereas the corpus callosum is very thin. But the relationship between those, either anatomically or developmentally, is not known at all. And it would be a very interesting animal model. For that, we will need to breed them in captivity to actually do experiments because it's very hard to capture them. They're protected and all of that. But it's an interesting case on how the ecology of the animal can actually provide clues on the anatomy and the evolution of the proportions and connections between these different areas. But yeah, that's a very interesting model because of that. It's one of the few animals that have such a massive ratio of hippocampal commissure versus callosal being very tiny.
Yes, thank you.
Welcome.
I have a question I think that might be relevant to the IRC5 about the contribution of the neocortex in different eutherian mammal species to the anterior commissure, because obviously all of the neocortex goes through the anterior commissure in marsupials, but it's a bit different in the eutherian case.
Yeah, so that's very interesting. Again, there's not too many studies, but there are some areas of the neocortex that do project down the anterior commissure, and they tend to be either immediately above the rhinal fissure near the insular cortex or the temporal areas. In rodents, they're not that well characterized, or they're not that big, they're not that prominent, right? There are some projections of the temporal areas and maybe a bit of the motor cortices that would project down the corpus callosum as well as anterior commissure. But in primates and carnivores such as cats and ferrets, there is more of a connection from the temporal lobe, right? So a lot of the temporal lobe and either some parts of the prefrontal cortex would also be projecting down the anterior commissure. What is not clear is whether those neurons would also project down the corpus callosum, and whether those would come from the same neurons that would be branching, sending one branch through the corpus callosum and another down the anterior commissure, or whether they would come from two different neurons, one projecting callosally and the other down the anterior commissure. But in the case of humans, that's also relevant because of this temporal lobe component, as well as the perirhinal and entorhinal regions that project down the anterior commissure. Another interesting area is the entorhinal cortex at the back of the brain, because that one is involved through all three commissures, and it's involved in connecting with the hippocampus. So there's also connection through the hippocampal commissure. It's also involved in neocortical circuits, so they also project down the corpus callosum, and with the olfactory circuits, so they would also project down the anterior commissure. So a cool thing is that if you inject in the entorhinal cortex, then you will see all three commissures labeled. Question would be again, are these coming from single neurons that branch, or are those different neurons that would take either of these routes?
And do you think if you could fuse somehow the midline of a marsupial with the midline zipperglia and those molecular mechanisms, do you think that any axons would cross?
Yeah, that's also an interesting question. I think that they might, but it may not be sufficient. Maybe if you could somehow delay SATB2 or also play with the timing of maturation at the same time, because one thing is to have the substrate and another thing is to have the axons already getting there. So if you could combine both production of the midline substrate with forcing axons to get towards there, it's very likely that these axons will cross. Because in preliminary observations, we found that the Danard, they also have glial populations that are equivalent to the glial wedge and the inducium gricium glia that are kind of channeling the callosal axons to cross through. And these glial cells also seem to be producing chemorepellents because axons avoid those neurons. You can see axons going in between the neurons. So probably if you have some sort of fusion artificially that you could promote in a marsupial, then I would expect to, yes, see something similar to a corpus callosum. That would be the $1 million paper, though, to have a Corpus callosum being made in a marsupial.
Great. Is there any other questions for Rodrigo? Let me know in chat if you'd like to ask a question or you'd like me to turn on your mic. No. Okay, well, we're almost at the hour, so if there's no more questions, with that, I'd like to thank everybody for coming. It's been a great attendance for this session, and also thank very much Rodrigo Suarez for all of his time putting together this presentation on closely evolution. So Rodrigo's contact details are there, and he's made the kind offer of answering any further questions anyone might have afterwards. And hope you're all looking forward to the March IRC5 webinar. We'll put out some flyers about that, about our speaker closer to the date. And hope you all have a great month and are staying safe wherever you are in the world. And hope to see you soon. Thanks, everyone.
Thank you.
Thank you.
Bye.